Hello first year students, we are starting with a new practical and the heading of the practical is Metacheomatic granule staining. Here we are going for the technique which is called Albert's method. Now in this method we require curd, we require chloroform which is already filled up in the jar, and this is our chloroform bottle, you can see very nicely. Okay, it is already filled in this jar. Then we require Albert stain. You can see this is our Albert stain. Can you see it clearly? Okay. Now, and the next one is Lugol's iodine solution. This is our Lugol's iodine solution. So, with the help of these three things, we are going to stain the metachromatic granules found in the bacteria which are found in the curd. Okay. So now first of all we will take the curd and it will be little diluted with the water, and we will take the very clean type of the slide, okay, which is already sterilized. Still we will sterilize again, okay, for our confirmation. Then here we are not making the round to show the smear because we are going to then after use the chloroform, so it will be mixed up with the chloroform. Okay, the material of glass marker will give the coloration. Okay, so it will go away, so we are not using here the any type of marker pen. Okay, like this we are making the smear. Okay, little amount of curd is taken and with the help of little amount of water we are making the smear. Can you see the smear? It will be little dirty white in color. Can you see the smear? Okay. Now we will again sterilize the nichrome wire, and our slide is then after kept for a drying purpose. Okay, so we will keep it here for make it air dried. Okay, now by the time we have already chloroform with us, and we have to keep it in the chloroform for 2 minutes, so it will remove the fat which is found in the curd totally. And then after we will apply Albert stain for 7 minutes, and we will apply the heat from the bottom, okay, with the help of burner. And then after we will again cool it down properly for 3 minutes, and then one wash with distilled water is given. Then after we will apply Lugol's iodine for 1 minute, and after that we will finally wash the slide and observe it in the microscope by using 100x, that is oil immersion lens. Here we are going to see the observation in this staining, that we will see the granules, metachromatic granules, brown in color, and the bacterial cells will be green in color. Okay. So now I think our slide is already air dried. You can confirm it by checking that now there is no water drop inside, already dried properly. Now after this drying we will keep this slide in the chloroform jar. Okay, now before you keep it in chloroform jar, you have to confirm that what is your true side. Okay, true side means where you have made your smear. Okay, so you have to remember. So here I am keeping the slide for fat removal for 2 minutes. Okay, then after we will have Albert stain. Now after completion of 2 minutes we will keep it out, and then after Albert stain is applied. Now here on wire gauze I will keep my slide and apply Albert stain. It should entirely cover the smear. Okay, can you see the smear? Fine. Now I will entirely cover the smear with the Albert stain. Okay. And now here I have to keep the wire gauze before I will give the direct heat. Okay, so direct heat should not be given. The slide should be placed over the wire gauze. And now I will arrange my burner in such a way that I will give the heat to the entire smear properly. Okay, so now I will little make the burner slow. Okay, I will make it little slow. And apply the heat. If it cannot be a lesson, then you can remove it for after intermittent heating. Okay, so that can also be done. So whatever the thing you can do, you can. Okay, so now I will again start the burner. But it should be having very little flame. Okay, like this. Okay, so this is enough. Now I will give the heat to the smear. Okay, continuously like this for 7 minutes. You have to count the 7 minutes perfectly, and after 7 minutes allow to cool the slide properly. Okay, you can see the thing like this. Okay, here our slide is there and it is being heated from the below. Okay, like this. Now if you see that your material is boiling and okay, too much of the precipitation is being done, then you can add your Albertstein again. Okay, you have to check. So now I think that I should add some more Albertstein, because it otherwise it will be precipitated out and I am not going to see the bacteria. Okay, because of the precipitates of the dye molecules. So now I will make the temperature little less with the help of burner. Now I am giving the heat continuously. Okay, so this way for 7 minutes I will do, and then after I will allow the slide to cool down properly. Okay, and then after I will give the wash. Okay, wash is to be given in the same way which we have given in the spirochete staining, with the help of distilled water. Allow it to cool properly. Okay, after it will become properly cooled, we will drain the material like this, and then after, with the help of little flow, very gentle flow of distilled water, we will try to remove the stain. Okay, like this. So stain is already removed. Now keep it again, and the next stage is application of Lugol's iodine for 1 minute. So we will add the Lugol's iodine. So now I am applying Lugol's iodine for 1 minute. Okay, the smear is completely flooded. Allow it to react for 1 minute, and then after we will wash it with distilled water again. Then the microscopy will be done. Here you require color pens for the figure of this metachromatic granule staining, you require brown pencil and also green pencil. Okay, so I will show you with the this sketch pens, because otherwise the color pens will be very light if I am going to show it to you just now. Okay, so now we will wash the slide again with little gentle flow of water. First of all, we'll drain the material, and now with very gentle flow, okay, it is to be washed properly with distilled water. And now I will air dry this particular slide of metachromatic granule staining, and then after we will do the microscopy by using 100x. Okay, so I am keeping it for air drying. And now when we are observing it through the microscope, what is to be seen? Okay, so first of all I will write down the name of the method, okay, on the top of the table. So I will write like metachromatic granule staining. Metachromatic granule staining, and the method is Albert's method. Albert's method. Now here we will make two columns in this table. First column is of figure and another column is of morphology. Now first of all we will take the green color pen and we will try to draw the bacteria, which will be usually rod in shape like this. Like this, okay. And now inside this bacteria the granules will be found in the number of 2 or 4. Okay, it may be usually 2, 4, 2, 3, 4, okay, depending on the species of the bacteria, okay, or even the age of the bacteria, or we can say nourishment of the bacteria, like this. Okay, this is our metachromatic granule staining. You can see very nicely. Okay, fine. If I will draw it in a bigger way, it will be like this. I mean, in more magnified way. This is our bacteria. Okay. And inside this bacteria, the granules are found. These are granules. Okay, can you see them clearly? Okay, so now we'll write down the result in morphology column, that the organisms are rod shaped, showing brown granules inside their cells. The bacteria are stained with green color. Okay, so this is our result of Albert's method for metachromatic granule staining. Okay, thank you very much.